So up top, I'm going to do a couple of reminders, kind of get to know ADDF. 26 years young, founded by the Lauder family, Ronald and Leonard Lauder, in memory and honor of Estee Lauder. At the beginning of when they were meeting with Howard as a co-founder with the two brothers, when they thought about what are we going to fund in Alzheimer's, they decided on a couple of things. Only translational science. What does that mean? Translational, how do you go from the bench to the bedside, right? So how do you take a novel idea and somehow turn that into a treatment or a support for people with Alzheimer's? Second, there would be three pillars of scientific funding. Biomarkers, which we're going to hear a lot about today, prevention, and therapeutics. Three, they were not going to give out grants. Rather, they were going to make investments. We're what is called a venture philanthropy, which means if we're going to put money into an academic idea or money into a biotech idea, there is a contract. And if that idea goes on to any kind of commercial success, the ADDF gets a portion of that to put immediately back into the science. And 26 years ago, this was a revolutionary concept brought to you by Leonard and Ronald Lauder and our very own Howard Phillip. The other principle at the start of the organization is actually where you all come in. And this was Leonard and Ronald saying, even with their wealth and influence, they cannot do this alone. And anyone who had an interest in translational science and the three pillars in this venture philanthropy investment idea could work with the ADDF. And again, that's where all of you come in. We have a couple of claims to fame in that time, over the time. About 10, 15 years ago, Howard got a call from the University of Pennsylvania from a scientist who said, I have an idea that we can do a trace of a dye that can say you have plaques and tangles in your brain. Why was that important? Because pretty much up until then, the only way you could see these telltale plaques and tangles, which is you've got Alzheimer's, was in an autopsy. And the idea was, could there be a PET scan? Right? And Howard funded this out of the University of Pennsylvania. It got spun out to its own company, and that company ended up getting bought by Eli Lilly. It is the Amyvid PET scan, and right now that PET scan, which is reimbursable now, 75% of the market of PET scans for Alzheimer's in the United States is that very PET scan. More recently, I think this was back in 2020, the ADDF funded the first blood test to come to market to tell you, are you at risk for Alzheimer's? That blood test is represented here on our panel today. Joel, raise your hand for a second. It's Joel's blood test. We're going to hear a lot more about that today. First to market. Okay. The last kind of claim to fame that I'll go is a belief Howard has always had when it comes to Alzheimer's, and that is the biology of aging. And to give you kind of a story or context around that, about 10 years ago, our good friends from pharma, and first of all, I want to very much thank both Biogen and Lilly who are sponsoring this symposium today. And Biogen and Lilly, along with the pharmaceutical company Eisai, they got interested in this concept that if you have plaques and tangles in your brain, if you could remove them, you'd be able to improve cognition. And when pharma went all in on that, and I want you to think about it again, because ADDF, here's what we do. Novel, interesting ideas, that's a molecule. We get them into the clinic, into humans. Phase 1, are they safe? Phase 2, are these ideas connecting, right? Target engagement. And the idea is we fund through phase 1, phase 2. Pharma comes in at phase 3 to take it across the finish line. So the money that you give goes into that, what is often called valley of death, where really unique ideas left unattended will go to die. We see them through with your help. So when pharma went all in on this plaques and tangles idea, Howard pivoted. And he pivoted to this biology of aging, that plaques and tangles is one of the signs of aging, but there are these other pathways to aging: genetics, inflammation, vascular, or mitochondrial systems. And I hate to tell us all here, but as we all sit here today, every one of us is aging. And we all age differently based on those characteristics I just went through. So in the last 10 years, Howard pivoted to where the light's not shining, right? And we've been like a swift boat, and all we're doing, shots on goal, because the board asked Howard at the time, hey, do you think it's more inflammation? Do you think it's more mitochondrial? Should we go all in on genetics? And Howard said, I don't know, that's science. We need to go across all those things. So we've been doing this for the last 10 years. We have an active portfolio through your funding of over $200 million in active portfolio trials going on right now. 85% of our funding is in clinical trials. The other 15% is in these late stage preclinical, that's the last step to get them from animal models into humans, right? And again, phase 1 is about safety, phase 2 is about target engagement. And truth be told, we're actually funding a couple of phase 3 studies right now because they're not quite ready for pharma to come in and take them over. We will not leave them if the science is good, right? We feel like we have this obligation to continue, if it's good science, to keep it alive. So that's what we do. All right, the architect of all of that, the aforementioned Dr. Howard Phillip. And Howard's been doing this 45 years plus, although I think the math last night was even a little higher than that, Howard, so there we go. Howard, it's been 2 years since we've been together, right? Many of us are just back from this incredible meeting in Madrid, the CTAD, Clinical Trials Alzheimer's Disease Conference. So much breaking these days, biomarkers, prevention, therapeutics, combined therapies. Can you just give folks, kind of last 2 years an update? Where are we in the field?
Well, I've been going to these conferences for, 30 years or so. And most of the time when you go to the conferences, it's science, presentations. There's not a lot of optimism or enthusiasm for what's going on because everything's preclinical and, we want to get to having drugs for patients. But the last couple of years, and especially this year, it was actually really exciting because research on Alzheimer's disease really didn't start till about 1980. Research on aging started more than 120 years ago. Cancer Trust and the Imperial Cancer Trust in England was started about 1905. So we're in a time frame really kind of shifted about 50, 60 years from other diseases like diabetes, hypertension, and cancer, which are diseases of aging. And one of the reasons for that is that although Alzheimer described a case of Alzheimer's disease in a 52-year-old woman in 1906, using the same dye that is now part of the PET scan, really, and the disease was described publicly in Austria and European conferences then, nothing was done to research this disease. It wasn't even, senility wasn't even recognized as a disease of old age. It was thought to be a normal part of aging. So what happened was in 1968, 3 pathologists working in London did a very simple experiment. They took 50, 75-year-olds at autopsy, stained their brains with the same dye that Alzheimer used in 1906, and divided them up into 2 groups. One group that died with senility, which was thought to be a normal part of aging, and another group that died who weren't senile. And it turned out that the old people, the 75-year-olds, who died who were senile, had plaques and tangles. So it wasn't until 1968, 1970, that that senility was recognized as a disease of old age as opposed to being a normal part of aging. So to give you that perspective, I could tell you one more thing in perspective. I went to medical school in 1970, 74, never heard of Alzheimer's disease, wasn't in my textbooks, never diagnosed a patient with it till 1980, when I walked into a nursing home and saw 300 people who were bedridden and dying from Alzheimer's disease, and recognized that this was going to be a big problem. The National Institute on Aging was started by one of my mentors in 1975, and the purpose of that was to jumpstart research on Alzheimer's disease. So when he went there was the time when there was a war on cancer, a war on heart disease, billions of dollars in 1975 being spent, and as a nation we spent $625,000 on research in Alzheimer's disease. None of it was really therapeutics at all. The reason I give you that perspective is Here it is, it's about 40 years later, and we finally, for the first time, have drugs that are disease-modifying agents on the market. And it actually was estimated in some modeling studies that from the time of a new research finding to the translation of that into a drug that's therapeutic is about 35 years. So although we're time shifted from these other diseases of aging, the mechanisms are similar, and we're kind of right on time in terms of the amount of research that we've done. So this last meeting, in terms of biomarkers and in terms of therapeutics that we have, not just the new monoclonal antibodies that attack the amyloid, which was one of the most obvious targets because it's part of the pathology, but our approach for many years, actually more than 10 years, has been from almost the beginning that aging is the leading risk factor for Alzheimer's disease. But there was no cross-fertilization, there was no crosstalk between the many, many, many scientists who are gerontologists who are studying aging for 100 years, and the neurology community, essentially, that was trying to develop drugs based on the pathology of Alzheimer's disease. And as Mark mentioned, this includes inflammation and genetic changes that occur every day, oxidative problems and misfolded proteins, vascular contributions. And so our portfolio has been very diverse. And what happened about 20 years ago was that the initial findings about amyloid, the genetics, the observation that it might be a drug target, it got adopted by Big Pharma. And Big Pharma has spent billions and billions of dollars bringing these drugs to market. When the focus of research in industry went to beta amyloid, there wasn't really a need for venture money, for venture philanthropy money anymore. It wasn't our role. We weren't going to really be a drop in the bucket to what pharma was doing. And so that gave us the opportunity to really focus on all these other mechanisms. I will say that that the portfolio, there's been an ongoing study for many years now about what the portfolio looks like worldwide for therapeutics for Alzheimer's. There's about between 120 and 140 different drugs being developed for Alzheimer's disease. And about 5 years ago, about 75% of the drugs in development were anti-amyloid drugs, predominantly, again, in industry. And today, only about 25% of the drugs in development are anti-amyloid, and 75% of the portfolio are on these equivalent of anti-aging mechanisms, as I talked about already, inflammation and so on. So there's been a shift worldwide in saying, okay, we got a handle on beta amyloid, we got a couple of drugs to market, we're going to need better ways to do it, got to go from infusions to subcutaneous administration, find regimens that are safer for patients. But there's more to it. We've recognized that these drugs only slow it down by about 35%, which is a big number. About 40% of people will remain stable for a year or two. And so there's clearly advantages in efficacy in these drugs, and safety like everything else. But it's clear we want to get to 100%, and not just to 100% in treatment. We want to get to 100% prevention. We want to delay the onset of symptoms from Alzheimer's disease to a point where it's been studied that if we could delay the onset of symptoms by 5 years, we would reduce the number of cases worldwide by 50%. And I do believe that's an achievable goal. What's going to make all of this happen, and what made it happen was, Mark mentioned the amyloid scan that we started funding 25 years ago. The first disease-modifying drug that was approved by the FDA for Alzheimer's, they said that because these new drugs remove the amyloid from the brain, and you could see that on the PET scan that we helped to develop, that it was reasonably likely, that's a regulatory term from the FDA, it was reasonably likely that that biomarker, which is a neuroimaging biomarker, would result in a clinical benefit. So the whole idea of that example is that the reason why our mission statement from the very beginning has included biomarkers, treatments, and prevention. Biomarkers are critical to drug development. Imagine trying to develop a drug for heart disease if we didn't have the biomarker cholesterol and we couldn't measure the cholesterol in the blood to see if statins were actually working. Imagine trying to develop a drug for hypertension if we didn't have a biomarker of a blood pressure cuff. We couldn't do it. It would be impossible. Imagine even trying to develop drugs for cancer. It used to be we'd see a solid tumor on your x-ray in your lung. Now we can take cells and type them and characterize them and tailor through precision medicine to your cancer, regardless of what organ it's in, and then give you drugs to tailor your biomarker abnormality. So biomarkers are critical to drug development. They're also critical to prevention, because now what we can do with blood tests like Joel is developing, and of course many others that you'll hear about today, is that to do prevention, we have to identify people in the preclinical phase. And prevention methods to test people, like cholesterol, like blood pressure, they have to be really inexpensive, because we screen large populations at scale. And we can't do that with expensive tests like neuroimaging, which cost $8,000 a ton.
And Harry, that's a good place for me to, let's jump in and introduce the panel, because we're going to get right to that.
Okay.
The panel today is all about diagnostics, and I think many of you know ADDF's commitment to this. We have something called the Diagnostics Accelerator. This was the partnership between Leonard Lauder and Bill Gates, and it has folks like Jeff Bezos giving money to it. His Biogen gives money to it. Lilly gives money to it. And what's interesting about that is that in addition to that, the two other panelists today represent companies who have received money from the Diagnostic Accelerator. And again, just this, we have a PET scan, but drugs are coming to market. Is there now a more efficient and less invasive way to get people into trials, onto the drugs, monitor them on the drugs, and even get them off the drugs? And we just came back from this conference, and I can tell you that it's kind of 2024, it's the year of blood. There are all these blood tests that are starting to come to market, C2N, Fuji-Ray Bio, AllsPath. It's literally like becoming like a land grab. But beyond blood, there are things like CSF, alpha-synuclein, optical, digital, right? And today, we have an expert panel to get into all of that with you. I will just introduce the panel, and then I'm going to come into questions for the panel. We start with, to my left, Danielle Graham. She's head of biomarkers at Biogen. So from an industry perspective, how does this look? Then we have, next to her is Joel Braunstein, the CEO of C2N. They were the first to market, and now there's a whole bunch of these blood tests, and Howard was saying last night, the NIH recently had a scientific bake-off to see what was the best test out there of 8 or 9 of these tests, and guess what? C2N ended up in first place. Congratulations, we're number 1. But not to be outdone, next to him is Russ Leibowitz. He's the CEO of Amprion, and Amprion has the first test for CSF. And this is a test that goes beyond plaques and tangles, and it has application to things like Parkinson's and Lewy body dementia. So it's really kind of an all-star lineup here. And let me start with you, Joel. First of all, what I really want to ask is, how does it feel to be number 1? You can answer that if you want. But in addition to that, or maybe most importantly, Joel, the C2N test, what makes that test so effective?
Well, first of all, I want to thank everyone for showing up today, and this is a real privilege and honor to be able to participate in such an incredible panel and important topics to discuss. I guess the first question and comment regarding the NIH study. So, there's nothing potentially more stressful than being put into a 9 tests round robin when you're the CEO of one of those companies. And C2N Diagnostics has really spent a lot of its history in trying to map and understand some of these important biological mediators of Alzheimer's disease and related forms of neurodegeneration. And people say, well, C2N Diagnostics has been around for a while. 2007 is when we formed. But I don't think we'd be I know that we would not be where we are today without that investment of time, resource, effort, commitment from groups like ADDF, who actually was our first venture philanthropy support effort back in 2007. Howard had the vision to see the importance of the technology that we were developing to say, This will help elucidate some critical questions that we have to understand how Alzheimer's disease originates, and then thinking about how we can develop practical solutions to ultimately being able to identify patients who have existing symptoms, concerns for cognitive impairment, to be able to accurately diagnose patients better than the current standard, which 15 years ago was really a clinical evaluation and detailed neuropsychological testing, maybe a couple of imaging studies, and even as recently as about 3 months ago, in a paper from JAMA that we were involved with, that standard of care has really not evolved that much. And we say, well, we've got these incredible tools like CSF testing and amyloid PET, but we know some of the challenges of access to those modalities. And when you look at organizations and clinical centers that are committed to using biomarkers to better diagnose Alzheimer's disease, they actually only use them in about 15% of people in the best case scenario. And then I always ask the question, coming from the field of cardiology, where that field has a lot of its success in the development of cardiovascular therapies that have had a profound impact on reducing mortality, reducing the comorbidity associated with heart attacks, really is based on the availability of tools. The first lipid-lowering drug was approved on the basis of LDL lowering. And so that analogy that we heard with regard to the use of amyloid PET and the amazing work that Biogen did to be able to demonstrate you can use amyloid PET assessments to have a likely clinical benefit based on what we're observing in the biomarkers is really an astounding accomplishment. And then I say, well, this makes where we are today an extremely exciting time because we're right at the beginning to be able to use these tools to now guide effective drug development and prevention strategies. I'll stop there. I could obviously keep going on, and we have other people who can contribute, but that's a little bit of the perspective.
I actually have
where we are
about
today.
8 follow-up questions, but we'll come back to that.
Thank you.
Russ, Alzheimer's, it's multifactorial. It's just not about the plaques and tangles. There's a lot more going on. We have co-pathology, Alzheimer's-related dementias, FTD, Lewy body, crossover to Parkinson's. Your test, CSF, alpha-synuclein. Let's start with, first of all, what are CSF and alpha-synuclein, and how does this relate to your work of being the first CSF test?
Sure. Thanks, Mark. And before I start, I want to echo what Joel said. Really, nothing we're talking about today would have happened, would be known without ADDF and everyone here. And it's also a real unique pleasure to be on this panel. ADDF funds people at all stages of development, but on this panel are people who've actually produced products, taken them through the regulatory process, and they're available now as real tools for diagnosis. That's an unusual feat, and I salute my fellow panel members. It's not a trivial thing. So to answer your question, Mark, CSF, cerebrospinal fluid, is a biological fluid. It surrounds the brain, mainly on the outside, in some places in the center of the brain. It cushions the brain, but it is intimately in contact with the brain, so it reflects what's going on in the brain very, very intimately. So you have things that are, if there's something that's damaging, it's easy to detect in CSF. CSF also holds some of the nutrients. The CSF passes 2 or 3 or 4 times a day into the blood, so there's a connection there. However, not everything in the CSF passes to the blood when we're talking about biomarkers. You have certain things that are very short-lived, and they may be very important, but after an hour or so, they disappear. They can be detected in the CSF, and getting to CSF is more invasive than blood, but much less invasive than something like a biopsy. It's something that can be done routinely. And so it's a very, very important tool to access what is going on in the brain now. The question then is, what's there that might reflect these diseases? And so I want to talk a little bit about alpha-synuclein. All of the neurodegenerative diseases seem to share something, that they are diseases reflecting misfolded proteins. So normal proteins in brain cells that have a normal function, under certain circumstances, change their shape, they stick together, and they form structures that actually are capable of replicating and spreading throughout the brain. So once this happens, through the CSF very frequently, this is how a disease spreads throughout the brain and gets to be something that has damaged many cells that we might later call Alzheimer's or Parkinson's. And synuclein was originally actually identified as part of plaque in Alzheimer's, but when people looked in more detail, synuclein became very closely associated with Parkinson's. Almost every case of Parkinson's had synuclein in cells in the midbrain. Also with a related disease called Lewy body dementia. And we believed originally that all of these were separate, that Alzheimer's was a disease of amyloid and tau misfolding. Parkinson's, Lewy body dementia were diseases of synuclein misfolding. ALS and frontotemporal dementia were diseases of a protein called TDP43 misfolding. But what we've learned from the tools that Joel and Danielle and my company, Amprion, has developed is that these are much more common, that these diseases bleed into one another. And so we're very pleased that we have the first test for looking in CSF for the actual misfolded protein itself. So let me say that there are two types of biomarkers. They are equally important, but one is a reflection of the disease. The other is the cause of the disease. It is the HIV to AIDS. And the reason that's important is if you have a biomarker that is the disease itself, you can hold it up, you can look at it, you can look at its structure, you can develop drugs against the disease. So both types of biomarkers are very important. What we work on at Amprion and with synuclein is actually the particle that causes the disease. And we amplify these. That's why it's Amprion, and the type of particles with misfolded proteins are called prions. The original disease for this was mad cow disease, and the discovery that a protein particle could spread and replicate basically led to a Nobel Prize for a scientist at the University of California. We now have taken that concept and understand that it is also the basis of all the major neurodegenerative diseases. So we can look and amplify from a single patient, and the structures here will be different for every patient, and the combination of misfolded proteins will be different. So we are enabling an era of personalized medicine, which was pioneered for cancer. It applies every bit as much for neurodegenerative diseases.
Yeah, well done, Russ. And again, for us to think about this, and I heard Howard years ago describe it this way: the plaques and tangles are like the scabs, and what's causing the scabs underneath. And we work on both of those things, right? And ultimately, it's about prevention, combination therapies, and precision medicine, which spectacularly is a lot closer than you think. 2 years ago, we weren't even mentioning these concepts in this room, so it's great. Danielle, I'm going to come to you, but I have to do a disclaimer first, and this is about, I think you all know we're honoring Biogen today, and it's interesting when I hear Biogen as part of big pharma, because I think probably Biogen thinks about scrappy, innovative biotech, cool, out of Boston, right? And there are a lot of firsts on this stage, so I need to point out some firsts. Biogen, the first to have accelerated approval by the FDA. Biogen and working with Eisai, the first full FDA-approved drug, lecanemab. So a lot of firsts on this panel, right? That we go. Danielle, I know you, like many in this room, it's not just a professional vision for you all of this, it's also personal. Like so many of us, we know this disease up close and personal in our families. But really, in your professional life, at Biogen, you and your team, we've heard there's PET scans, there's blood, there's CSF, How do you, from an industry point of view now, how do you look at those three things? Are they all important? Is one more important than the other? Is one going to replace another? Walk us through that from an industry perspective.
Sure. Thanks, Mark. Happy to. And similar to my co-panelists, before I start and answer your question, I just want to say thank you. Thank you again to you, Mark, the entire ADDF, for the opportunity to be here. It's an absolute privilege. But thank you to you all. I mean, it's an exciting time to be in Alzheimer's disease because we are seeing such tremendous advancements, but there's still a lot more work to do, and it's because of the support through you all and the ADDF that we're able to see such great advancements. So again, thank you. So Mark, you asked about CSF, blood, and PET, and how we think about that from an industry perspective, both in the context of clinical trials, but also in the context of clinical practice. Before I answer how we think about it today, I do want to also share that one of the reasons why we have seen such success with the new therapies on the market today is because Biogen was one of the first to use amyloid PET, so the test that Mark described that allows you to look at the amyloid burden in the brain, in their clinical trial for aducanumab. And by doing so, by including only those patients that actually have the amyloid pathology in your brain, we were able to show that there was, in fact, a reduction of that amyloid pathology and then clinical benefit. And this was a trial that was done years ago. And as a result of that success, all of the other clinical trials started to leverage amyloid PET and only include those patients that had the amyloid burden to be able to demonstrate a benefit. And so we know amyloid PET is by far one of the critical tools that we do need to utilize to diagnose Alzheimer's disease. But as a result of the work that others have done, we can tell you that CSF tests, similar to what Russ has done with Amprion, CSF tests that allow you to look at amyloid in your CSF are highly similar to amyloid PET. So today, in the trials that we enroll, we are primarily using amyloid PET and CSF, or CSF, not both, to confirm amyloid pathology. The most exciting thing, however, is the work that Joel and others have done to demonstrate that you can actually use a blood test, a much less invasive test, to demonstrate this amyloid pathology. In terms of clinical practice, we are not using blood tests to replace PET or CSF at this time. There's still some more work that's required, but the inclusion of these blood tests in our clinical trials has dramatically changed our efficiency and our cost. We're able to reduce the amount of time it takes for a patient to get into a trial, as well as reduce the cost associated with putting patients through PET scans or CSF that don't actually require it. So it has changed the efficiency of our clinical trials. And then, of course, moving to clinical practice. So you mentioned lecanemab, and of course, Lilly has donanemab as well. One of the biggest challenges that we face today is being able to adequately and accurately diagnose patients with Alzheimer's disease and that amyloid burden. And with the blood tests that Joel and others are doing, we have high confidence that by getting those tests out, that will enable patients to get onto therapies faster. So it's a really exciting time, especially from the blood-based perspective.
Yeah, phenomenal, Danielle, and that's going to get me in the second round of questions for you all. I just want to give you guys a preempt. I'm going to do one more set of questions up and down here, and then we're going to open it up to the audience. So there are microphones in the middle. So if you're thinking of questions, or be thinking of questions, because we'd love to take them from you. So Howard, that's really interesting, listening to the panel and where Danielle has just left this off. And I know, you're a neurologist, primary care physician, right? That you do see some patients, right? Concierge patients. And you've had patients, you've had them take the PET scan, you've had patients, you've had them take the blood test. How do you look at this, and not only for yourself as seeing patients of where this is evolving to, right? But as you look at primary care physicians across the U.S., geriatricians across the U.S., right? Do you see this, hey, tomorrow they're all going to be given the blood test, or it's 40 years from now? So starting basis of your own practice so people get a feel of that, and then project out for the other geriatricians and primary care physicians.
Yeah, so I remember the days when Alzheimer's was a clinical diagnosis. You kind of thought you got it right, people with cognitive impairment, but we didn't have any blood tests or spinal fluid tests to confirm the diagnosis. Then we evolved the PET scanning, and the spinal fluid test. But now we have these blood tests. And blood tests are part of the normal workflow of a doctor's office. You know, we have blood tests for heart disease and so on. So I think the blood test is going to be revolutionary.
Right.
Because it's going to move the early diagnosis of cognitive impairment and Alzheimer's into primary care.
Right.
And in other words, there have been studies that show that the average time of delay between first onset of symptoms and the time a person finally gets a diagnosis is 2 to 3 years. But now I envision a time when people will come into the primary care office, if they have some memory complaints, maybe they'll get some brief cognitive testing just to see where they really are. But then they'll get a blood test. And so that there won't be that delay, because those 2 or 3 years of delay can be really catastrophic for patients and their families when people have cognitive impairment and they're not diagnosed and treated and managed. And it's especially true now that we have these disease-modifying agents that are anti-amyloid agents. We want to know early as possible what the diagnosis is and if people are eligible to get these treatments. That's critical. And so I think that the blood tests are really going to revolutionize not only diagnosis, but access to treatment.
Yeah.
And that's going to be really important. And the other thing is we have, as Russ mentioned, alpha-synuclein and beta-amyloid tests now on the market. We're seeing some movement in TDP-43, as was mentioned, and of course we can't eliminate tau. But what we're seeing now, and I think we're coming to recognize, is 40% of people with Alzheimer's disease, as defined by amyloid tangles, also have alpha-synuclein in their brain. So as was mentioned, we used to silo these pathologies. Now we're seeing that everybody's got a mixed dementia, probably. So I think that the future is going to be in diagnosing people with these mixed causes, because I think Joel was telling me a little earlier that people that have both beta amyloid and alpha-synuclein are much more rapid progressors. Their diseases are more rapidly progressive illness. So that's really the future of how these diagnoses are going to be panels. Inflammation is really important. We have one blood test on the market now called GFAP for inflammation, but we need, Danielle and I were talking about this, we need much better blood tests for measuring inflammation in the systemic circulation as it's reflected in the brain. So there's a lot more work to be done, but it's a really exciting time because now for the first time we can get a definitive diagnosis in the primary care setting.
Thank you, Harry. Joel, so this is an interesting question for you. Here you have this phenomenal #1 test. Give folks a sense of, I know you're now already starting to work with the Mayo Clinic. I know you're also working in countries like Japan, about your test, kind of the rollout of this test.
Thank you. So 2020, we introduced the first blood test, and that was called the Procivity AD blood test. And I think we surprised a lot of people. We kind of kept it low profile until we made the announcement. And the next thing I know, I received a call from AP News to say, tell me about this blood test. This is going to drive a lot of consumer interest. And I tried to explain what we were doing, and we were very careful in how we rolled it out, because we said there's a lot of implications to having a test that can now provide a quote-unquote pathological assessment of Alzheimer's disease. Historically, Alzheimer's disease has been diagnosed clinically, as Howard already talked about. And so this concept of trying to redefine what Alzheimer's disease means is the same way that we came to characterize and come up with much better approaches to cancer care. And getting into staging and getting into precise identification of what pathology exists enables one to now have that much higher level of certainty. So when we rolled out the Procivity AD test, overall it was a very positive article, but there were a couple of comments because media loves drumming up the pro and the cons. And there were some comments by some experts in the field say, this is too early. Are we getting ahead of where the evidence is? And the question always becomes like, when is the right time to introduce a disruptive technology like this? And sometimes perfection is enemy of the good. What we found with that test was we never launched a very aggressive campaign to advertise it because we said this needs to exist in the hands of skilled memory care specialists. We need to let them get used to using the test. We need to continue publishing important data. And C2N has been involved with over 200 global collaborations and hundreds of thousands of analyte measurements and test measurements to understand how to use these tests in clinical care. And so with that effort, of course, has been increased trust, increased recognition that these tests are really as robust as one may proclaim based on an early set of promising data that you see in the lab. We just introduced the Procivity82 test about 9 months ago, and that was a step forward compared to the first generation test. That new test, Procivity82, included measurements in the blood that looked at both amyloid particles that are a key component of amyloid plaques and also tau particles. And tau is also a really critical protein that's implicated in the clinical progression of Alzheimer's disease, and in fact is possibly the highly relevant species to be able to stage to understand where a person is in the pathological process of Alzheimer's disease. And so as we've rolled it out, we've seen an incredible amount of both consumer interest and clinician interest, but we continue to work primarily with memory care specialists because we think they're the ones that are going to help shape this new standard of care. And then what we find in the community is when neurologists are comfortable using the test, they're the ones who are frequently being approached by the primary care clinicians saying, well, I have a patient with a memory concern. I'm reading more and more about all the advances in Alzheimer's disease. Help me think about the best approaches. And with that, we've been able to really make incredible inroads within the US. We have several thousand clinicians that are currently ordering the test. Unfortunately, the test is available in 49 states. You say, well, which state is the outlier? And here we are, New York. New York has a special certification process that can often take years and years before testing becomes available. The good news is we're working through that process, but you don't get a lot of feedback as to exactly when that certification will occur, but we do expect it to occur soon. So the good news is that this is now an accessible technology. And then what's happened is as we've been able to have very compelling clinical evidence to show these tests are as accurate as CSF and PET. And we've established that in multiple different studies, culminating with the recent publication that made international news in JAMA. There was a paper several months ago picked up by the Wall Street Journal, New York Times. Actually, over 500 global media outlets picked it up. And it was basically a test using our test in over 1,000 people in both primary care and specialty care to show that you can actually raise the clinical diagnostic accuracy from 60 to 70% with the standard of care evaluation tools. So we talked earlier, clinical examination, a detailed neuropsychological assessment, maybe some imaging studies and routine blood tests. You can raise that diagnostic accuracy from 60 to 70% up to over 90%. And that is a transformation of clinical care. And we will see that happen more and more with more studies as these blood tests become more routine. And as Mark alluded, we currently offer the test through direct access with C2N Diagnostics, and we recently partnered with Mayo Clinical Laboratories, which is one of the largest clinical reference labs within the United States. They put a high premium on quality, so we've partnered with them to offer access for those patients that work through the Mayo Clinic Labs network of several thousand hospitals. And then we've also partnered with a very large reference lab out of Europe for 75 countries. We offer testing in Brazil, Australia, and Japan increasingly. So I think you're going to start to see this roll out over time, and it's obviously very exciting.
And I love to say it also soon to be Brooklyn and Staten Island and Queens
Yeah,
and the
absolutely,
Bronx and Manhattan, which is good. Russ, you you talk about a scan. Do you ever see a day that you're looking at a blood test and it brings in things like TDP, H3? So can you comment? There's a difficulty in doing that for what you're looking at, but I know you're looking at it.
Well,
So
it's a great question. What we look at, we can measure in different tissues and fluids. Right now, as I said, we're looking at the particle itself, the infectious particle, and it is, even in spinal fluid next to the brain, we are assessing that it's there with less than 100 molecules. That is an unthinkable number. There are no tools that can detect 100 molecules. So what our technology does is it amplifies it 1,000,000-fold, 10,000,000-fold, until you have enough that you can really look at and analyze. So it requires amplification. At least right now, when that gets passed on to the blood from the CSF, it means that in a mil of plasma, there might be 1 to 5 particles. And right now, we don't know that. We can't detect them. It may be that they get filtered out, but obviously it is a high priority to be able to do that. But right now, what we've learned by being able to look even in the spinal fluid, which, as I said, is more invasive, but it's just a needle. And for many years, everyone who has
just
multiple
a pinprick, huh?
everyone who has multiple sclerosis, of which there are a lot of people, you cannot get your treatment until you have a small amount of spinal fluid. Joel has and colleagues have set the standard that it is possible for some of these to see in the blood. Hopefully we'll get there, but if not, we have access. And let's talk about what that access means. We don't, for Parkinson's disease, for Lewy body dementia, for some of the other molecules, the other misfolded proteins, we don't have a PET scan. It's very interesting in itself because amyloid and tau were fairly susceptible to molecules that bind that you could see on PET. So we believe, for the same reason I've told you that even in CSF there's less in the brain by mass and not enough to see even by PET. So we have to take the tools that we have. But given the tools we have, the studies we've done have been in many ways an epiphany. So what we have learned is that using our test, going back to what Joel has said about the difficulty of clinical diagnosis, even in the hands of the world's best doctors, 30% of patients diagnosed as Parkinson's in an NIH database, so it's all medical centers using the best protocols, don't have any synuclein. They don't have classical Parkinson's. And when we look at autopsy, it matches our test exactly rather than the clinical diagnosis. 30% of Lewy body dementia, which people believe is one of the easier to diagnose because you have so many different symptoms that come together, don't have synuclein. And then in a recent study that really we were introduced to by ADDF, we looked at the entire database of a study called ADNI, which is the longest running Alzheimer's study with imaging data. And there, they made sure from the very beginning that if anyone had signs that looked like Parkinson's or Lewy body dementia, they eliminated them. They excluded them. And yet, in their study, with our test, 40% of those people with Alzheimer's, having excluded any other disease clinically, still have synuclein. And we're now trying to learn with our partners, some of whom are in the audience and on the stage here, if those 40% will respond the same to these magnificent drugs that we now have out there. We don't know, but we're going to learn soon because of tests like Joel's, the drugs that Biogen and Lilly and Eisai have produced, and our test. We're going to learn if those fast progressors can be slowed down or whether we're going to need a combination therapy for that group. So I think we're making incredible inroads in understanding. And, blood, it would be great if we could do the same thing there, but we're learning so much and we're affecting the course of the disease that we'll take what the biology gives us.
Fabulous. Danielle, I'm going to give you the last word here with a particular question, but then we're going to open it up to the audience. And again, we have microphones in front and back, so feel free to line up as you see fit. Danielle, I'm going to come back to the blood test, and I was fascinated by the conversation you had about how important that blood test is in the clinical trials. Take us to that place where Howard's talking about primary care physicians with that blood test and the ability with that blood test to start thinking for the drugs that you and ASI and Lilly are doing going earlier. And just explain to people how from an industry point of view, you think about that.
And Mark, when you're saying going earlier, are you talking about going earlier in the patient journey? So earlier in the stage of disease?
Exactly right. Of how we have these drugs now for there are people for mild cognitive impairment,
right.
but between the treatments and the blood test, it kind of opens up a window. Can we think about this earlier?
It's a great question, and it's one that we and others are thinking about quite extensively. Many studies have shown that in a lot of different indications, the earlier you initiate treatment, the greater clinical benefit, the greater improvement on memory and cognition you will achieve. And so what we're seeing with some of the blood tests that Joel and others have developed is that you can actually start. Now, this is still in some of the research stages. It hasn't been actually applied to clinical practice. The hope is that this will be available in clinical practice soon, is that the earlier that you administer or run these tests, the earlier you might be able to identify a patient that's headed down the Alzheimer's disease journey, and then the earlier that they could be initiating treatment. And of course, the intent and the excitement there is that the earlier these patients are on drug, the more likely they are to show even greater clinical benefit. Now, again, we're still generating some of that data, but that is the exciting aspect of where the blood tests are.
Thank you, Danielle. And this is where we're going to open it up to questions from the audience. But I asked that question because it's so interesting the times we're in. And for the folks that have been with us for past fall luncheons, I hope you get a great sense of pride of hearing these kind of things may be coming, but they're here now. And what's happening, because how many times, Howard, have we heard, well, I don't even know how I could find out if I'm at risk, and why would I even want to know? And not only are these first drugs coming from pharma reasons, well, you can do something about it, but the way pharma is looking at these drugs is, can you get them even into people earlier before there are any symptoms to push out, push out, push out? And Howard, you've given this stat before about average lifespan and then how much we can delay the onset of Alzheimer's. Can you just go over those numbers again?
Well, the average age of onset of clinical symptoms with Alzheimer's disease is about 75.
Yep.
And the average age of death is, let's say, just for argument's sake, about 80.
Yep.
So the idea is that if we could delay the onset of symptoms, which is the main goal, I would say, by 5 years, then the average person would have the opportunity to die of a heart attack in peace in the middle of the night, instead of spending somewhere between 5 and 9 years suffering loss of mind, which is, as we all know, kind of a horrible thing.
Yeah. And just on because, as we all die of the heart attacks, and bless us all, but think about that. No cognitive loss. Knowing your kids, your grandkids, all of us who've seen this disease that takes away humanity a week at a time, a month at a time, years at a time, not having to think about that anymore. And you're hearing this here. This is not pie in the sky. This is what's in front of us. Do we have questions from I know there's one. Go right. Please, and just line up at the microphones and we'll take your questions as you go. That's a little tall, isn't it, Carlin?
Yeah, this is all very exciting, but if you're looking at personalized medicine, how does genetics play into how you're going to personalize this? Because I gather that some of the genetics that predispose to Alzheimer's also make the treatments more dangerous for people. And are there ways to screen people who have genetics sooner that with the blood tests and everything, and perhaps do what you're talking about, and it would be safer to treat them earlier? Or I just wondered.
I'm going to let two of you answer that. We're going to start with Joel.
Yeah, so genetics obviously is a critically important factor and variable that can influence predisposition to a condition like Alzheimer's disease. The genetics carries a lot of weight to it, right, when you provide genetic information, and particularly for individuals that are asymptomatic. So there is a clear use case that didn't actually exist prior to 2 years ago. The use case is to consider, let's say, an apolipoprotein E genetic analysis to understand one's predisposition perhaps to a side effect from some of the anti-amyloid drugs that are now emerging. That's an extremely strong use case because you can use that marker, the genetic risk factor, to identify perhaps predisposition to the side effect. And that's starting to lean towards personalized approaches. The question of being able to use it for identifying people at high risk before they've developed symptoms, and this is a whole separate conversation.
Yeah.
Well, a lot of my discussion today is really focused on what Danielle was referring to, where it's like early MCI, early mild to moderate Alzheimer's disease with symptoms. Now the question is going towards prevention. That is where we want to go, and that I am highly confident we will get there. There are several organizations in this audience who are leading major international studies on prevention, looking at people that may be deemed quote unquote high risk, and that high risk can be determined based on some of those genetic markers. But you also alluded to this point briefly, that these genetic markers are not deterministic, they're probabilistic, meaning you can only use them to identify, let's say, the risk. They don't guarantee that the disease will be present. And that's where the power of having specific markers that are indicative of pathology, let's say the early biochemical abnormalities that are indicative of an Alzheimer's disease pathophysiological process happening. And those are some of the measurements that our team and other groups are innovating. And it is impressive. We can look at individuals 20 years before the onset of symptoms and begin to see abnormal signatures in the blood that are highly indicative of who may be progressing or on this trajectory to ultimately develop clinical Alzheimer's disease. The moment you can identify those people precisely, and our team, that is a major, major effort, it requires hundreds of thousands of individuals being studied because of the diversity of people throughout the world to understand how to interpret these markers. But the moment you can identify, have a reliable set of tools to identify that quote unquote high risk individual, then you can start thinking about very effective strategies, some of them therapeutic, pharmacologic, others lifestyle behavioral modification. And I think ultimately, and we've talked a little bit about combination approaches, combination therapies. This Alzheimer's disease, the brain tissue is not so different from cardiovascular tissue and other tissues throughout the body. And so we understand complex diseases that are really diseases associated with aging. There are a whole variety of factors and pathways that are contributing to the ultimate clinical presentation. And it will be a multimodal approach. Just like treating heart failure requires not just a single agent, but multiple drugs to improve in lifestyle modifications to really improve that patient's longevity and quality of life.
There's several people I like to go on the panel on this one, but I want to make sure we get to everybody's questions also, so we may come back to this. Why don't we go to the next question?
Thank you. My question may be similar to the genetic factor you just discussed, but what is, or do you know the percentage, if your parent had Alzheimer's, the percentage of children having the disease?
Howard, I'm going to toss that to you.
Yeah, family risk, if you have one first-degree relative, parent or sibling, who has Alzheimer's, your risk is about 5 times the normal population. If you have 2, it goes up to about 15.
Thank you.
Okay. We have a question that's in the back, so we're going front and back of the room, all the way to the back of the room.
Can you hear me? Does this work? It's working? Oh,
Yeah.
Great. I am so encouraged by what I'm hearing, and I have a question. If these tests and identification systems would work with someone who has a comorbidity of cancer,
I didn't get the question.
Sorry, would you
You're going to
repeat
need that a little bit louder. It's a little harder to hear.
Oh, I'm sorry.
Yeah.
Can these tests work if someone has a comorbidity of cancer?
No. Who wants it? Danielle.
I'll take a stab at it, but I'd love to hear Joel's perspective as well. It's a very important question, and if you think specifically about the blood tests that we're talking about and that are becoming available, it's still new, it's still early, but we are starting to see some data, and this was just shared at Madrid at the CTAD meeting, that there are some indications. One is actually kidney failure or kidney, sorry, it's not kidney disease, but kidney indications that can influence the levels of some of these blood tests. That doesn't mean that if you have cancer that you wouldn't necessarily be able to use the test. It's just as you are taking the test, it needs to be part of the diagnostic workup, and that information needs to be included. I don't know if Joel wants to add anything else to that.
Yeah, I think that's a complex question. Clearly, there are many systemic factors. Cancer maybe is a comorbidity or cardiovascular disease or renal impairment that can alter the circulating levels of these markers that are indicative of brain pathology. It impacts not only blood, but also CSF. And so there are certain analytical techniques that one can use to ensure that you've normalized and can avoid the confounding effects of, let's say, a condition like cancer or kidney impairment. It's one of the analytical methodologies that we use. We believe our technique actually is equally robust across different comorbidities. This will be a critical issue to really understanding implementation of blood biomarkers, broadly speaking, in primary care. Primary care, and we demonstrated this in the JAMA paper, has considerably higher amount of comorbidity in the patients who present to primary care compared to specialists. If a patient is self-referring to a specialist, they're pretty focused on a condition, or maybe they're of a certain socioeconomic group that enables access to the specialist quicker. But the reality is that the majority of patients presenting with these concerns come to primary care. And as the primary gatekeepers, they are not only concerned about the patient's cognitive condition, but also other comorbidities. So they tend to see more heart failure. They tend to see more people with hyperlipidemia, more people with kidney disease. And so really understanding how these biomarkers perform in that context is going to be critically important. Some of our early data is extremely encouraging that, at least with our methodology, one can not be too concerned about the confounding effects.
And Howard, and I don't want to break any HIPAA laws here, but have you ever experienced that in your practice, somebody with cancer coming in?
Yeah, of course, cancer is very common in old age, and many patients will have cancer and then Alzheimer's disease or cognitive impairment. Another twist to that whole thing is chemo brain. A lot of patients that undergo chemotherapy will have cognitive. We've tried to figure out how we might get involved in chemo brain as a risk factor and as a cause of cognitive impairment. So it's Alzheimer's disease and related dementias. Chemo brain is usually self-limited in time, but it certainly exists, and it's a problem for families and for patients, obviously. So I think it's the oncology world didn't really want to accept it for a long time, but now I think it's come mainstream that chemo brain is a prominent cause of cognitive impairment in older people.
Thank you. Next question, please.
Thanks. I have actually two questions for Russ about the...
Just a little bit closer to the mic, if you could.
Sorry, two questions for Russ on the Amprion, the CSF test. It seems to me that it kind of covers all bases with the amyloid as well as the tau as well as the alpha-synuclein, being able to test all of those components. I'm wondering, how available is that type of test? We were talking about the Precivity 82 is only available in 49 states. Is this a test that you have to have sort of the cognitive assessment first, and then you can say, I'd like now this CSF test? And I know there's intrathecal, so it's a little bit more difficult. And then my second question, there are many rare diseases that actually show as a biomarker an aggregate of alpha-synuclein. Are you doing this test at all in pediatrics or for rare diseases, or are you thinking about that?
Great questions. I'll address what we know. So for synuclein, it's available, and it is the one test that right now is validated against many, many patients and against autopsies. And it is very accurate at all stages. Like Joel has described for some of the blood tests, we know that we can follow people, we can find the first moment when they turn positive, but what I will tell you is most people, it is very rare to have a positive, so there's almost no background. We haven't looked in particular pediatric cases, but I'm not aware of synuclein. Well, obviously, if there are mutations in synuclein, you can have this early. But then there's a gene that tells you that. We deal mainly, in many ways, people who are most interested and who benefit most from our test are those who have a genetic predisposition, whereas maybe 5% of people with that mutation will ultimately develop the disease, and they want to know. And we know that we can identify that 5%, and the reason is that when we look at people who have the genetic predisposition at about age 50, who have no symptoms at all, approximately 5 to 7% are positive. As soon as they have any symptom at all, and the symptoms for synucleinopathies can be as remote from Parkinson's and Lewy body dementia as losing your sense of smell. If someone has lost their sense of smell or have certain sleep disorders that still precede the disease by 20 years, instead of 5%, it's closer to 90% of those are positive. So we're really dealing, largely our data is in the adult population, people at risk. What you ask about people who would have childhood diseases, that's just harder to do. It always works best if something can be done first in adults and you prove it, then you get permission to look in children. And certainly we are very interested in any opportunities where we can help people, like everyone else here. We only benefit when we help people, and that is our main goal. Does that answer, or do you have?
Nope, that's good. Thank you.
Okay, thank you.
Please.
Thank you. Thank you very much for being here today, and thank you for the work you're doing, especially on the biomarkers. A question again for you, Russ, is you mentioned that misfolded proteins seem to be the link to neurodegenerative diseases. How can one prevent misfolded proteins? It can't just be genetic. The gene has to be turned on or off. And then, Howard, you were mentioning inflammation. So you think about Alzheimer's, and it's really the third diabetes and inflammation. So my question is, how do you prevent the misfolded proteins, and is that really lifestyle modifications?
Yeah, that's
And
the
Russ,
7
we're
billion people question.
Yes.
What starts it? And at least for synuclein, we have some ideas. As I said, you can have a genetic predisposition, but that makes up only, at most, 10 to 15% of the disease. So most has to be related to one of two things, and I'll tell you the data, and we can think about it together. One is aging, right? As we age, our ability, all of the proteins that misfold in the brain tend to be very abundant. They have a normal function, but they're present in relatively large amounts. So as we are young, we process them properly, we make a lot, we degrade a lot, there's a balance. As we get older, certain things fall apart. And one of the things that seems to fall apart is our ability to process some of these proteins. And as the concentration gets higher, because we're not processing them, we're making maybe just a tiny bit more than we're getting rid of, then they accumulate. And at higher concentrations, when they not only misfold, but the proteins, the misfolded form is very unstable unless it hits another. At a higher concentration, the probability of forming these misfolded aggregates goes up a lot. So part of it is aging, and I think we have data for that. And in fact, we know that when we look at weak genetic links to these diseases, to all of them, a lot of them are genes that control the, excuse me, the way that we process proteins inside of the cell. And even though they don't cause the disease, certain genes that might affect our ability to process seem to be linked at a very slightly higher probability of disease. So aging is one. The other is environmental. And the question is what's the hard data for that? Well, it's still speculative, but places where misfolded synuclein seem to start in the brain look like there are two sources. One, from the gut, because in the gut you can have misfolded synuclein, and it may pass along the vagus nerve into a part of the brain stem, and we see that that's an early place where people get disease. The other place where we seem to see very early signs comes through our nose, our sense of smell. In both our gut and in our nose, we're seeing all sorts of chemicals all of the time. And so it seems likely that a significant amount of these diseases are caused either by some combination of aging plus something in the environment, and we really can't identify.
Let me jump in because we want to make sure we get to questions. We're also going to refer you to, particularly as it relates to inflammation, you're going to want to talk to Howard when this is over, but I want to get to the other two questions.
So
okay?
thank you very much.
You have, once we're done, have shot at all of them to ask your questions.
Okay.
And the reason why I want to get to the next two questions after you is because I have a board of governor of ADDF and a board of overseer of the ADDF, and then I'm going to have to cut it off because we're going to be late for lunch. So Bonnie, start us.
Thank you for this great presentation. I was just going to ask, and you really answered it in a way, you know, is it just luck that some people get it and some people don't? Because I have know people in their 90s who are as sharp as can be, and I have friends in their 60s who have dementia, and my mother had Alzheimer's. So what is it?
I mean, I think any of my colleagues could handle that.
Yeah,
Most
Howard,
of
take
these
us-
are
go ahead.
sporadic. Howard's
and then
probably
we're
the
going to have
expert.
fast answers because now we got a third person there, so go.
Well, there's a lot of interest now in this question of resilience and what makes some people resilient to the ravages of aging in the brain versus others. And I think that part of that comes from a process called neuroprotection. We all might be exposed to the same oxidants and so on that can damage proteins, become misfolded, toxic. And it may be that some people have better systems of managing oxidative processes so the free radicals don't damage proteins to the same degree. But there's a lot of interest in your question right now scientifically in the science world.
Great. I want to applaud Dave because I was just going to call out all the males in the room. There hasn't been a male to ask a question yet, so good for you, Dave.
Well, you may be sorry that I asked that question.
Next.
Because it may be a silly question. My understanding is that the new drugs that have come out are only effective if they're used at a very early stage. And my question is, how early a stage do you have to be in before the drugs are effective? Once you become any kind of symptomatic, does that mean that you can't use the drugs?
So, Danielle, you want to take first crack or you want to give it to Howard?
Go ahead.
Well, I was just going to start by saying the drugs that are approved in symptomatic individuals. So these are individuals that do display the symptoms associated with Alzheimer's disease. So starting with that, because I think you were asking if it was only for those individuals without symptoms, if I understood your question correctly. So it is in individuals with symptoms. And it is...
It can be used with individuals with symptoms or not?
Yes,
Yeah.
that is correct. Howard, do you want to add anything?
What was the last thing you said? I didn't hear it.
No, it has to be the early stages of symptoms.
But there's a stage of the disease before dementia called mild cognitive impairment. And that's when people have memory problems, but not really any functional loss, and that was part of the trials. So people that just have memory problems would be eligible for treatment. And then beyond that, there's people that can have a positive brain scan, positive PET scan, without symptoms, which would be in the prevention stage of the disease. And I think, we hope that the prevention trials will work ultimately, so people who have almost no symptoms might actually, who have the disease, but no symptoms, could also be treated. But that might be futuristic at this point.
That's correct. It's not quite approved for individuals that don't have, that are in that amyloid positive, or they have the pathology in the brain, but no symptoms in that stage. It's not approved for that. That's one of many of the trials that are ongoing now to interrogate that. Joel, one?
Yeah, I would just add one other brief comment. I think over the next 5 to 10 years, this is what you're going to be seeing a lot of research in advance with regard to understanding stage of disease and this concept of personalized medicine. There are markers emerging, and our team is developing some of these markers that will allow us to very precisely stage the disease from a pathophysiology perspective, or the biochemical changes that follow this continuum, all the way from the earliest signs of some abnormality of the biochemical processing of these markers through late stage Alzheimer's disease with true clinical symptoms. And that's going to involve understanding the profile of these biomarkers, when they become abnormal, what you actually call abnormal, because today what we call is someone being, let's say, amyloid positive. Frequently that's referring to someone who has amyloid imaging positivity. But the reality is that we can actually pick up abnormal amyloid processing 10 years before amyloid PET scans show. But the question is, what do you call that? And so I think you're going to be seeing a very, very close collaboration between biopharma and diagnostic companies to precisely stage the disease and be able to identify, the right patient at the right time with the right therapy. And that's really, the exciting.
And Howard, and sorry, Mark, just one last thing. I mean, the really important thing to also think about is the biomarkers is what's changing the face of Alzheimer's disease treatment. And understanding that progression of amyloid and tau and the other markers that we're talking about is absolutely critical to think about the treatment. But the one thing too is that there is often, and Howard, maybe you can even comment on this, individuals who are amyloid, that have amyloid burden in the brain, that may not go on to develop Alzheimer's disease. So it's another thing that we have to think about as we're thinking about who to treat and who not to treat.
Good.
Thank you.
Last question. And Dave, you did great.
Tough act to follow. Thank you. You discussed the environment of the gut and the nose being possible environmental factors. My question is the cerebrospinal fluid that envelops the brain in any way the actual impact, the genetics of that fluid? Everyone has obviously different genetics, and is it the fluid of that genetics that could impact Alzheimer's? And then when those proteins do fold, is there a way to alter the DNA of those proteins to keep them from folding and replicating? And does that have anything to do with respect to the aging factor, the telomeres getting shorter?
Howard, give it a whack.
So 2 questions. One about the spinal fluid. The spinal fluid is kind of a system that washes the brain. And with aging, the flow slows down. So we've actually invested in a company at Stanford a number of years ago to try to have an implant, a device that would increase flow to get rid of the waste products that the spinal fluid is trying to remove. So that's one kind of answer to your question. The other thing is, with aging, these misfolded proteins are poorly managed. And to prevent them from misfolding, through evolution, the body cells have emerged, have developed a process called chaperone-mediated autophagy, which means that there are molecules that bind to these proteins to try to keep them in their proper state. And when that fails, the molecule goes down the line into what's called the lysosome. And we've been funding projects to both try to enhance chaperone-mediated autophagy to prevent the misfolding. But once the proteins are misfolded and they're toxic, they go into this organelle, this part of the cell, where it's a very acidic environment. There's lots of enzymes, and they digest the misfolded proteins, so they don't get out of the cell and start causing all kinds of damage. And at Stanford, we're investing in an academic program there to develop new drugs that enhance the acidification of the lysosome to enhance autophagy in patients with Alzheimer's disease.
Wow. Thank you.
Great. let me do this because we're getting to the time of lunch. I have a few words to wrap up, but first, just logistics. We're going to proceed to lunch in a minute here. The lunch, just as a preview, hosted by Emmy Award-winning Paula Zahn. The Lauder family will be there en masse, and I just remind everybody the Lauder family underwrites every single piece of this so that every cent of every dollar you give goes directly to the science you're hearing about today. We are honoring, as you've heard already, Biogen, but we're also honoring the Goodnow family and their foundation in memory of their patriarch, Ned. It's going to be a very special luncheon, what we used to call must-see TV, right? So, which is great. I really want to thank Joel and Russ and Danielle and obviously Howard, also Biogen and Lily for making this all possible today. And I want to emphasize something because there is never enough time to get into everything here. But this is moving now to prevention, democratizing diagnostics and blood tests, combined therapies, precision medicine. And what I can absolutely promise you, every dollar you give going forward, it is to speed that better prevention, earlier and more sophisticated, more sensitive diagnostics, combined therapies, and precision medicine. And for those of you who've been with us for the better part of the 14 years, like, wow, right? And for those of you who are coming in now, what a great time to be coming in. This is moving fast. Your capital can speed the day. So thank you. See you all at lunch. Oh, how about the panel? The panel.